Hello, and welcome to demola. And today's video, we are talking about classification, something that comes up basically every single year when we're talking about A-level biology exams. So let's make sure we cover it well. No time to waste. Let's get into it.
When it comes to science, there's always a reason for what we do. And in terms of classification, there are approximately 8.7 million different species on Earth, right? Massive number. And we need a way to organize them all into groups to help us identify species, etc., etc. And that is the reason why we have classification. So, studying classification is what we call taxonomy. So, it's the science of naming, describing, and grouping organisms. And when we have these different groups of organisms, we need to make sure that they're distinct, they're non-overlapping, and they're hierarchical. Cool. So, we'll get into what that means, but first of all, important one, kind of a throwback, we need to be able to define this word species, right? And I won't write it out here because it's a bit long, but species is a group of organisms that can interbreed to produce fertile offspring. Very, very important definition in biology. So we've got our taxonomy, and we know that our different groups are called taxons. So I write that down. The different groups are called taxons. Now, these acronyms are so important because you need to memorize this whole thing, and you need to memorize in order. So, because of like scientific developments, which we'll touch into a bit in the end, we've actually changed the system. So before we didn't have this top column domain. So that's new. However, what we need to know is the order. So I use the acronym King Prawn Crackers or Fat Greasy Sausages. Now, if you look online, I'm sure you'll find different mnemonics if this one doesn't suit for you, but this is the one I've always used. And it's literally just looking at the first letter and then applying it to the groups. So we need to know we're going domain, kingdom, phylum, class, order, family, genus, species. Where species is the most specific and domains is kind of general. And what we also use to know is you've got 3 domains. So eukaryota, prokaryota, archaea. Now, eukaryotic prokaryota, you already know. Archaea, these are like your bacteria that live in like these extreme conditions, so like in volcanoes, etc., etc. I would say you don't need to know much about them, but that's pretty much it. Very similar to bacterial cells, to be honest. Now, naming organisms is the last thing that we're going to cover on this slide. And we use a system called binomial nomenclature. So it's kind of reverse. So like if we do first name, surname, how like you and I are called, we actually flip it over. So it's last name and then first name. So we're going to do something more general, the genus, and then something more specific, the species. So for example, humans, Homo sapiens, we're going to say Homo is the genus, the last name. So anything else similar to a human is going to have Homo. However, the specific one that will identify us, okay, that's a human, sapien. So last name, first name, genus, species, Homo sapien. Hope that all makes sense. Okay, so now we're going to look at how we actually are going to classify organisms or say organisms are closely related to each other. Now, molecular phylogeny is what we use nowadays. But before, we used a traditional system where you were just looking at phenotype. So, important definition, again, is the observable characteristics of an organism. Yeah? We used to just say, oh, these animals both have wings, they must be closely related to each other. Oh, this animal's wing is pink and this one's is also pink, they must be closely related to each other. That's kind of the processes that they use to classify organisms before. But now modern systems more rely on the genotype or the combination of alleles present in the gene. Now, typically what they're going to use is amino acids, which is called proteomics, and we also have DNA or RNA sequences, which is called genomics. And make sure as well, bad on me for not writing it here, but we are talking specifically about the sequence. So if we have more similar sequences of amino acids or DNA and RNA, which means we're more closely related because we're going to make similar proteins. If we make similar proteins, we're going to get similar phenotypes. That's the whole kind of basis behind it. So your molecular phylogeny, we're using that molecular side, the proteomics, genomics, and we're linking it to studying evolutionary links between organisms. And that specifically comes through common ancestry. So what you'll typically see, and they can kind of do this in different formats, but you have a phylogenetic tree here. Now, analyzing this tree is really important. So we've got 5 different species, A, B, C, D, and E, right? What you'll see is which ones are closer together. So every single time you have a branching off, this is a common ancestor. So you can see the most recent common ancestor between A and B and C and D is kind of happening in the same place. So you can see that A and B are very closely related. C and D are also very closely related because they share a very recent common ancestor. If we take it a step back, you can see here, this is a common ancestor between A, B, C, and D. So all of these have this purple one as a common ancestor, right? And then in red, we have a common ancestor between A, B, C, D, and E. So this one, whenever you have a diagram, we call this the LUCA. or the last universal common ancestor. So that's a very important one to be able to notice. And then also just making sure you're looking at the tree. If they're more close together on the tree, they are more closely related. So make sure you're looking at where these places branch off. Okay. So the last thing to kind of mention is about the scientific community. I thought it wasn't enough to get a slide on its own. But essentially, if someone does a bit of research and they think, okay, I've got a theory, I want to disprove something that we kind of commonly believe, they need to do research, right? So they're going to do whatever experiments they need to do, collect that data, and then they need to publish it. So they've collected the data, analyzed the data, compared it to what we had before, and they publish it, right? They can do this in a scientific journal, in a scientific article, whatever. It means that other scientists are going to be able to see that information. Once they've read it and they thought, wow, this is interesting, they do a process called peer review. So what peer review is going to involve is they're going to see the experiments that someone's done and probably repeat it or maybe make minor changes, but essentially they want to do an experiment to see, are we going to get similar results? Are we going to be able to disprove this theory? Cool. And that's going to link onto ideas of validity and accuracy, which are two very, very misunderstood words amongst A-level students. So I'm going to try and define them. Validity is kind of saying that your results are representative. So when we say representative, we mean we need a big enough sample size, we need a good method, we need our control variables, everything like that. You need to make sure those results are representative. Meaning, validity is that your results are close to the true value. That will make my experiment valid. Now, accuracy is a bit different. Accuracy is just that the results of my experiment are close together. So say, for example, I do an experiment and all of my results are about 5 units. Yeah, all of my results are about 5 units. That means that my results are pretty accurate because I'm normally getting the same thing. However, say the true value is 10 units. Then my results of 5 units aren't valid, right? They are not representative of the true value. Hence, it's not valid, but they were very accurate because they were close together. So make sure that you can understand the difference between validity and accuracy when you're kind of looking at these scientific articles or anything like that. You won't be asked to do that in the A-level, but you might get a set of results and just say accuracy, validity. So just make sure that you can think of those ideas as well. Okay, so question time. So you can see all of them there. I'll zoom into all of them as well just so you get a better idea. But this one, you're saying, explain how the information in the table can show that archaea are more closely related to eukaryote than bacteria. Cool. Here, we're saying that trypsin is an enzyme, and you're looking at the phylogenetic tree to explain how trypsin molecule's molecular structure is going to be able to make that tree. And finally, you're looking at how does the scientific community evaluate new theories. So you've got a couple of questions there to try. Have a go, and we'll get with you. Okay, so I know I said that archaea are more similar to bacteria, right? But this question told you that archaea are more closer to eukaryote. So remember, always go by what the question said. But when you analyze the table, what you'll see is that in red are the differences between archaea and eukaryote. In orange is the similarities between archaea and bacteria, and in green is the similarities between archaea and eukaryote. And as you can see, just straight off the bat, there are so many more similarities between archaea and eukaryote on this table. So we've noted of that. That's the first thing that you want to say. So you can see here, there are more similarities between archaea and eukaryote. Fine. Now, what you can also say, just to emphasize the point, there are fewer similarities between archaea and bacteria. Again, good. Then what you want to say is literally, and whenever you have a table in biology, I think always just put examples because you're getting marks here for literally just writing on the table. So like, for example, saying that both of them don't have their growth inhibited by streptomycin, and both of them sometimes have a cell wall. Literally saying things like that gets you another mark. And then again, you're linking it back to this idea about the study of evolutionary links. You're saying that they must have evolved from a more recent common ancestor. That's how you get the marks there. Now, the next one was about using this trypsin molecule. So it says, explain how the primary structure of trypsin is used to produce a phylogenetic tree. So again, we're thinking, okay, primary structure, what we need to think, that's the sequence of amino acids joined by peptide bonds. So first of all, we need to know what is the sequence of amino acids. Then what we can say is, what are the differences or the number of differences in that sequence between species? And if there's more differences, we're saying they're less closely related. If there are more similarities, we're saying they're more closely related. But making that point again gets you those marks. Finally, when we're talking about peer review, First of all, they need to communicate the theory to the scientific community. So this is what we were saying. Scientific journals, conferences is another good one. Articles, whatever. It doesn't really matter. As long as you're getting that they're communicating it to the scientific community. Then we need to do a process of peer review. And then we need to make sure, okay, were these results actually valid? So are they true? Are they close to true values? Have they gone through the right processes in terms of control variables, etc., etc.? Making sure that's all in place will mean that we can start to think about that theory as true. So that's how you answer those questions. Okay, a bit of a short and sweet topic. So we're going to get into the A-star tips now. First of all, we need to understand, why do we need to classify organisms? That's the most important thing, first of all. There's a reason why we're doing this. The hierarchical system where we're going from domain, kingdom, whatever, make sure that you know that offhand. Like, I don't need to know, I've done my A-levels, but you guys need to know. So make sure that you learn that. Also, being able to do that binomial nomenclature, honestly, it's easy marks if you can do it properly. So make sure you can. And then molecular phylogeny is probably the harder bit about this topic, but making sure that you understand what it is, why it's used, and also about all that proteomics, genomics, and comparing sequences. Now, what do we need to make sure we're not doing? Not being able to interpret those phylogenetic trees. It's very important that you can see a tree and you can tell, okay, where are the common ancestors? What's the Luca? What is the more recent common ancestors between this species and that species? Make sure you're really comfortable doing that. Now, neglecting the importance of peer review in scientific theory. I'll be very honest with you. It's not a common question, especially in SNAB, but it's a question that I think is pretty easy to pick up marks on. So just make sure that you understand what's really going on there, because I don't think it takes too long to learn, even if it's unlikely to come up. And then finally as well, molecular phylogeny, you have to link that to topic 2. So when we're talking about like transcription, translation, we're talking about protein structure, they like to link those topics a lot. So that's something you need to be aware of, especially in SNAB when they're always pressing on about synoptic thinking. So thank you all for listening to that video. I hope it's been really helpful. If it was, please like and subscribe to the channel. We are getting very close to 1K subscribers, which would be amazing. If you have any questions about anything I've said or biology questions in general, drop them in the comments below and I will do my best to answer. We've also got our TikTok and our Instagram, both at A-star demola. Link should be in the description below. We're putting out great content, getting closer to exams on there now. And that is all for me. So thank you for listening. Hope it's helped, and I'll catch you in the next one. Bye for now.